Welcome to another episode from the Veterinary Research Channel. Today, we're diving deep into a fascinating study that scrutinizes the reliability of D-dimer assays across different animal species. Specifically, we will discuss how well the antibodies in these tests react to D-dimer in dogs, cats, and horses, and what this means for veterinary diagnostics. To begin, let's set the stage with a bit of background. D-dimer is created when the body breaks down blood clots. Measuring D-dimer levels can help diagnose thrombotic conditions like deep vein thrombosis or pulmonary thromboembolism. In veterinary medicine, these tests, primarily designed for humans, are often used on our animal companions. However, a significant question lingers. How accurate are these human-based assays when applied to different species? The central problem is that commercially available D-dimer assays rely on antibodies developed to target human D-dimer. The concern is that these antibodies may not react the same way with D-dimer from other species due to subtle differences in molecular structures. This can lead to inaccurate results, potentially affecting diagnosis and treatment decisions. To tackle this issue, researchers at the University of Melbourne conducted a detailed study. Their goal was to evaluate how well various commercially available antibodies against human D-dimer bind to D-dimer in dogs, horses, and cats. They hypothesized that there would be significant differences in how these antibodies react across species. To test this, the researchers collected plasma samples from healthy dogs and horses. Since obtaining surplus feline plasma was difficult, they used pooled plasma from cats that were submitted for diagnostic purposes. It should be noted that the feline pooled plasma came from cats with a variety of diseases. While not ideal, the aim of the study was to evaluate the immunoreactivity of D-dimer. The researchers then prepared cross-linked fibrin lysates from these plasma samples. This process mimics what happens when blood clots are broken down in the body. They used a method called SDS-PAGE, or sodium dodecyl sulfate polyacrylamide gel electrophoresis, and immunoblotting with several commercially available antibodies to human D-dimer. These techniques allowed them to visualize and assess how well the antibodies bound to D-dimer from each species. Several antibodies were used in the study: Anti-Fib, DD5, DD44, and D2D. These antibodies were selected to demonstrate the differences in performance and to emphasize the implications of using unproven assay kits. So, what did they find? The results revealed that the antibodies reacted differently with D-dimer from each species. For example, one monoclonal antibody, DD44, bound canine D-dimer with good specificity and sensitivity, but did not react with feline or equine D-dimer. On the other hand, the polyclonal antibody D2D bound putative D-dimer in dogs, cats, and horses with good specificity and higher sensitivity compared to human D-dimer. The anti-Fib antibody showed strong binding with fibrinogen in cats, dogs, and horses, and with human D-dimer. DD44 reacted strongly with D-dimer in dogs, with no bands detected in the plasma samples for any species. DD5 showed consistent reaction with fibrinogen in all species. D2D did not bind any of the higher molecular weight peptides, including fibrinogen, but demonstrated strong binding with a protein of similar molecular weight to human D-dimer in all species. These findings highlight a crucial point. The variability in performance of commercially available human D-dimer assays across different species is partly due to interspecies variation in D-dimer immunoreactivity. In simpler terms, what works for humans might not work as well for animals because their D-dimer molecules are slightly different. One of the most significant implications of this study is that veterinary professionals should be cautious when using human D-dimer assays on animals. The study underscores the need for validation studies to ensure the accuracy and reliability of these tests in each specific species. The researchers also noted that monoclonal antibody DD44 could be a promising candidate for developing a canine-specific assay. Given its high specificity and sensitivity for canine D-dimer, it could lead to more accurate diagnoses of thrombotic conditions in dogs. The study also sheds light on the challenges of diagnosing disseminated intravascular coagulation, or DIC, in animals. DIC is a complex condition involving abnormal blood clotting. The authors point out that D-dimer measurements do not reliably contribute to the diagnosis of DIC in cats. The presence of D-dimer in healthy horse plasma could be associated with subclinical disease. To explain the differences in immunoreactivity between species, the study suggests the most likely reason is the difference in the specific residues involved in the binding sites of each antibody. Future studies could evaluate differences in sequence and structure of D-dimer between humans, dogs, cats, and horses. In conclusion, This research emphasizes that while human D-dimer assays can be clinically useful in veterinary medicine, they should be used with caution. It is essential to recognize the potential for interspecies variation in D-dimer immunoreactivity and to perform thorough validation studies. Furthermore, the study identifies promising avenues for developing species-specific assays that could improve the accuracy of diagnosing thrombotic conditions in animals. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.